Okay. Yeah. Uh, Good morning. Good morning to our host, Dr. Preeti Kumar. Dr. Preeti Kumar is currently working as an associate professor with the in infectious diseases with the Yale School of Medicine, and she has very graciously agreed to do a short interview on a a subject which is very close and dear to everybody's heart in India right now: how to fight COVID-19. We are all, the whole world is under the pandemic, but fighting COVID-19 needs many things. Among them, the most important thing is a vaccine. Currently, we are having vaccines, but the whole world is facing vaccine shortage. There is possibility of developing vaccine some other way. We'll get more to know from Dr. Preeti Kumar. A little introduction to Dr. Kumar. Dr. Preeti had been in India earlier. She has done her PhD in Indian Institute of Sciences, Bangalore. Then later on, she went to USA, did her further studies in USA. She has also worked in Harvard University, and currently for last 7 years, she is working for Yale School of Medicine. Yale is, as you all know, a very prestigious university in America. And she has been instrumental in setting up a BSL 3 level lab which is known as Dr. Kumar's lab, which does research on various things, mostly into the viruses and their behavior and their genomics. And chiefly among them were AIDS virus and Nile virus. And she's engaged into research on those. Let me welcome you, Dr. Preeti, because most of the terms in your introduction sheet, I am not able to understand. I'm unfortunately an IT person and not a person of the genetics, although genetics also is a database and IT is also a database, but we only have 2 bits. You have, I think, 4 or 5. Anyway, shall we start now? Thank you very much for spending time for this. And it's morning with you. So good morning once again. And,
good morning, and thank you for having me.
Oh, it's our pleasure, entirely our pleasure. We hear a lot about something called a DNA vaccine. What is a DNA vaccine, and is it something different than what we have been getting injected all along here for COVID, I mean?
Yes, indeed. You used the key word a few minutes ago called genetics. Yeah. DNA is something that encodes genes. I mean, DNA is present in everybody, and that's the source of all the RNA and proteins that are made in humans. So every protein that you see in humans, viruses, animals, plants, every living being, essentially comes from something called nucleic acids. And one form of nucleic acids is DNA, or deoxyribonucleic acid. Our chromosomes in humans are made of DNA. And the way DNA acts is the DNA is, for people who know a little bit about the cell, humans, every cell in living organisms has got something called a nucleus. The DNA is present within the nucleus. The DNA is, is made into a molecule called RNA, which is ribonucleic acid. And this RNA is transported from the nucleus to the outside, which is called the cytoplasm in the cell. And in the cell, cytoplasm then makes proteins, which we use as building blocks in every cell and, to carry out every activity in life. So as opposed to the usual vaccines, a DNA vaccine is something where you put the original nucleic acid, DNA, inside a person's body. So this DNA will enter the cell. It's supposed to enter the cell, go to the nucleus, and now start producing proteins from, from DNA to RNA to protein. So it's actually higher up in the hierarchy. It's the founder or the father of what you get finally. Right. So that's a DNA vaccine. As opposed to this, every other vaccine that you've seen out there normally is a protein vaccine. It's either a killed viral form, you can take the virus and kill it, or you have an attenuated or weakened virus, which you can use the vaccine. That's been the traditional vaccine source used. But lately, for the past year, you'd have heard this word RNA vaccine, mRNA vaccine. Right. And I told you RNA is the intermediate between DNA and protein. So that's another way of getting proteins to get expressed. Instead of putting in the founder or the father, that's DNA, you actually use RNA, which is the intermediate, and you can get proteins from there. So DNA is, more upstream in how you would produce a protein.
Okay. So we are going into upstream in the actual introduction of the protein.
Yes.
Okay. But then how would this vaccine actually work in the human body?
So it's very interesting. Quite a while ago, about 3 decades ago, there was this very interesting observation done that you could just introduce naked DNA. DNA is one of the simplest things to make. You can grow these plasmid DNAs in E. coli, and they're recombinant DNA pieces. You can grow them to really large quantities. They're really, really stable. And all of genetic engineering and recombinant DNA technology till now has come about only because of the fact that you can have small pieces of DNA. For instance, any gene in the human body can be expressed in a small plasmid, and this can be grown, and you can look at the proteins that are produced in E. coli or mammalian cells by introducing this DNA into these cells. So it was very interesting to see that naked DNA, without requiring any other kind of vehicle, could enter cells and produce protein. So that was something that really excited people a long time ago. And they realized that instead of making the protein in huge quantities, which is really difficult, it's not easy to make protein in huge quantities, unlike DNA. So you can take naked DNA, put it on cells, and in fact, even put it on humans, force it into cells, and you can start expressing a protein. And that made it a very attractive molecule to really use for 2 different reasons. Number one, DNA is highly stable. It can be stored under a wide variety of conditions. It can last for a very long time. And it can be used and it can be grown to very, very high quantities to really meet the demand, which is what we need right now in the world, given the pandemic. So it's something that can be scaled up. It's extremely stable. And just naked DNA, you can, kind of inject into humans, and it will produce protein to make the protein that will be used for boosting immune responses inside the human body. So that's the advantage of a DNA vaccine. So when people found that this was possible before, there was about 2 decades ago, there was a huge rush towards making DNA vaccines. But of course, it didn't amount to much at the time. But coming back now, given the seriousness of this pandemic.
But you mean to say that now it is possible to manufacture this DNA vaccine, although maybe 8 years back it was not really very easy to make it? Does it work that way?
It was very easy to make it all the while. I mean, I must say that a decade back, it's one of the easiest things to do. It's one of the molecules that can be actually synthesized at GMP level. That means good manufacturing process. That's what is required. Can be done at a GMP stature extremely easily. It can be cleaned up. It's one of the hardest molecules to work with. The only difference was it was not as effective as protein vaccines. So when you use a protein or a viral-based vaccine, the kind of immunity you would get or the kind of protein expression you see in humans is a lot more. So really the efficacy of a protein or a live attenuated or a weakened or a dead virus-based vaccine was a lot more than a DNA vaccine, obviously because you're giving the last molecule directly, right? You're giving, when you're using a protein, you're giving a last molecule directly. But in the case of DNA, even though you can make it in large quantities, it has to go inside the cell, then go to the nucleus, then get transcribed into RNA, get translated into protein, and then produce a large quantity. So it's got multiple steps there. Even to enter the cell, it's not as easy as in the protein's case. A protein can just enter bloodstream and you will have an immune reaction. So that's the reason it didn't get very popular, because the efficacy was not as good as a protein vaccine. But manufacturing wise, it's one of the easiest things to do, and we have had the technology for a long time.
But is the efficacy getting better with the better techniques, or it is still in the same region? What is the range of efficacy it used to have?
So it's around the same, I would say. It's the same technologies that's being used even now as opposed to then. We worked on DNA vaccines for a very long time. So it was very, very easy to come up with a DNA vaccine now. The efficacy, I don't think, I can't say it would compare to what you have as protein vaccines or live virus vaccines, but in the face of a pandemic, where you have to really address the needs of billions of people, it's very, very difficult to make the other kinds of vaccines and very expensive too. So to really scale up and amp up an RNA-based vaccine or a protein-based vaccine to meet demand of the whole world, it's really hard. And you know, for instance, I could just give you an example right now. I think with the kind of protein vaccines that's available in India, you're targeting about 8 to 10% of the total population.
That's right. That's right.
So even if it was efficacious in 90 to 100% of the population, you're only protecting that small population.
Right. Okay.
On the other hand, if you have something like a DNA vaccine, which is easy to manufacture and can be, potentially administered to the entire 1.3 billion, even if it was 60% efficacious, you're hitting a large portion of the population. And that's where DNA vaccines, I think, have a major advantage in the scale of a pandemic like COVID-19.
Would it also help in creating the herd immunity much faster if we do that?
You see, herd immunity, again, depends on what kind of immune responses are elicited in humans. So herd immunity is when you're actually speaking, herd immunity is when you're naturally exposed to the virus. So herd immunity is a term that's used when you get natural virus infection and everybody becomes immune in the absence of vaccines.
So not with vaccine.
So the whole world was in fact hoping that you would get, there are administration, countries that still think herd immunity is possible if people get infected naturally and they become immune. But now it's well known with SARS-CoV-2 that even if you're infected once before, you can still get the virus again. So, and with all the variants coming in, you are not really protected from a new infection. So herd immunity is a little difficult to envisage in the case of SARS-CoV-2. So you need to pre-vaccinate to really even prevent infection or reduce the symptoms of infection. So with something like DNA vaccines, I would think you would at least target a large amount of the population, and get some level of immunity, which is badly needed in the absence of a large-scale manufacture of vaccines and availability of vaccines. DNA vaccine is something that one should definitely be looking at.
Okay. But does it mean that this DNA vaccine has been used before for some other diseases or other elements?
So there are two parts to that. DNA vaccines have been developed for many diseases, including cancers, but nothing has actually been licensed for human use. They've gone through several clinical trials. They've been found, safety has been proven. Nobody has ever seen any kind of a serious adverse effect. But nothing really became licensed for human use because there are much, much better RNA vaccines or RNA vaccines are also very new. RNA vaccines only started this past year with COVID. So in the light of the fact that several more efficacious vaccines were available, and number two, a pandemic of this size has never been seen when they were contained in small pockets. There was no need to really develop a vaccine that, or no need to really license a vaccine that had less proven efficacy because there was no demand for it. I mean, there was no unmet need for something like that. That being said, the licensed vaccines for DNA do exist for West Nile virus, but that's for use in horses and birds in the USA particularly. So you want something cheap to be able to vaccinate animals that really are the intermediate hosts for all these viruses. So why not a DNA vaccine? If you use a DNA vaccine and go to all the equine species or birds, you're not spending much, but you're actually immunizing the intermediate host that spread these to the human population. So approved licensed vaccines exist for animals and birds, but nothing got approved for humans so far.
But did they conduct any trials on humans?
Several. Absolutely. I mean, and they've all been, I know in India, there were major trials, in fact, conducted for rabies. So we do the DNA vaccine for rabies was, I think, on somewhere in the early 2000s, I think people were developing it. When I mean, it's about 2003 to 2009, I think there was a big surge in trying, boosting a DNA vaccine for rabies and several other viruses as well. Clinical trials have been conducted, particularly for cancer. Mostly, they've all been found to be extremely safe. But potency issues really have prevented these vaccines from coming to the limelight and getting licensed.
So still research is going on in this subject?
It died down a lot. I must tell you that it was never as active as it used to be with in the past decade or so, because it's been moving away towards something more like mRNA vaccines.
Right.
Because now there are methods to deliver mRNA. So mRNA or RNA vaccines, the reason why that did not become as popular as DNA, RNA vaccines are just as efficacious. The reason is RNA is extremely unstable compared to DNA.
Correct.
So RNA cannot be stored, but DNA can be stored, which is why DNA was used. But on the other hand, because of its instability, it is difficult to deliver RNA inside cells as well. But now we have extremely good, kind of nanoparticles and liposome and other kinds of methods by which RNA can be delivered very efficaciously inside cells, which is why RNA vaccines were very quick to be adapted for SARS-CoV-2. And in fact, this pandemic has given the platform for RNA vaccines to come up, and we tried all of a sudden in humans, something which should have taken several years again now came to the light because of this pandemic. So now looking at the demand, I think DNA vaccines also would find light of day.
Okay. In fact, you said that it's a very stable vaccine. So for a country like India, where we have far-flung corners to deliver the vaccine, transport itself will be much easier with a DNA rather than an mRNA vaccine.
Absolutely. So mRNA vaccines require freezing at -70 or
exactly.
Something like that. DNA vaccines are stable at normal in the normal refrigerator at 2 to 8 degrees.
Okay.
In fact, there are studies showing that you can keep DNA in solution even at 25 degrees Celsius and nothing happens to it. As long as it's in water that doesn't contain nucleases, it's absolutely fine. RNases are highly unstable and require very special conditions for storage, which is also one of the reasons why RNA vaccines aren't that popular in India, given you can't take it, like you said, in and around everywhere to every rural area. On the other hand, DNA vaccines have that potential because they can be stored on ice packs, essentially.
Correct. Would there be any side effects of this DNA vaccine when it is developed?
So that's an excellent question you ask. The one reason why RNA vaccines didn't become popular for a very long time is they stimulate an innate immune response. So they're highly inflammatory. So they themselves stimulate an inflammatory pathway inside cells. So the minute you inject with an RNA, you have some kind of an inflammation developing. Now, in a weird way, that inflammation also helps boosting of immune responses. So if you have an additional inflammation pathway going on at the time when you prime an immune response with a vaccine, it boosts the immune response. But if this inflammatory reaction, it also gets seriously out of control in certain individuals, depending on the reaction, it could be very toxic too for the individual. So until recently, there was no way to reduce the inflammatory responses induced, non-specifically by these RNA vaccines. But in the past couple of years, they've discovered multiple ways to reduce the inflammatory profile of these RNA vaccines and make them a lot safer, which is why now RNA vaccines are in vogue. On the other hand, DNA is, while it does induce some level of inflammatory pathways, it's not as immunostimulatory as RNAs. So it's a lot more stable, and many clinical trials have pretty much shown that the adverse effects associated with these vaccines are very minimal to mild and really last maybe for a few, couple of days. And it's nothing to be worried about. For the large part, they're very safe and tolerated.
Okay. In the development process of a DNA vaccine, we have reached till what level? Like, my question is, when will it be really available for the masses for using?
So that's a difficult question to answer, but I can tell you based on what I know with the COVID vaccine that's being manufactured. So I read up a little bit. It looks like they've applied for emergency approval, and that happens only when you're capable of really delivering to a large population. So it's one of the easiest things to grow under GMP practices. And I know that places like Bharat Biotech in India have previously tried kind of vamping up DNA vaccines. It's something that can be done very easily for a country, for a company here, I would think. So what they hope to is, I mean, from what is being said from the company, that they hope to, to vaccinate at least crores of people on a daily basis, which is possible given the way you can actually vamp up these DNA vaccines. And they hope that in about 6 to 8 months, the entire country will be vaccinated. And with any other vaccine, such claims I would be worried about, because it's not easy to make them or store them. Correct. But with the DNA vaccine, I mean, I would think that at least part of what's being said is true. And the second important thing with respect to a DNA vaccine, I must say, is you don't need to use needles and syringes. There's something called a gene gun, which you just use to high pressure, just inject it inside a person. It just is like a little shot of a gene gun that you give. It just goes, penetrates the dermal, just the skin. It's intradermal. It doesn't even need to go inside. So there is no worry of using needles, contaminated needles, and going from person to person, making solutions, drawing it up in a syringe. You can load it up in something like this high pressured device, and just kind of make it hit the skin, and it'll go as a high powered blast. So the contamination is also exceedingly low. So the rate at which you can vaccinate people must be very high.
So in other words, actually, it is without breaking the skin. So there is no blood coming out.
Not puncturing the skin, but I'm sure it does, cause abrasions or a break on the skin. Otherwise, the DNA can't enter. It will not puncture the skin, or it will not lead to a blood wound. So it's exceedingly safe that way.
More hygienic, definitely.
Yes, absolutely.
More hygienic. Now, the critical question about every vaccine, how would it cope with the variants? Because that's the danger which people are facing currently. The Delta variant, and God knows more Greek letters are coming in.
Right. I mean, that's an excellent question that you ask. And when companies report a particular level of success against variants, right? I mean, they say that it's 90% efficacious against the main variant or the Wuhan strain, but it is only 60% successful against the Delta strain or the Lambda strain. The way they measure it is by taking the antibodies from the vaccinees and looking at how these antibodies protect against other viruses. But what is not measured is, in addition to antibody-based responses, there are a lot of other responses which are called cell-mediated immune responses. There are cells in the body called T cells, and T cells also really play a very important role in protecting against any of these viruses. What these T cells do, while antibodies can go and bind the virus and stop the virus from infecting, the T cells can kill the cells that are infected with the virus. So not only are you targeting the virus, but also the source in which the virus is replicating and producing more. So nobody looks at the CMI response that a vaccine induces. And in fact, together, the CMI as well as the antibodies contribute a lot towards protection. And even though it's reported as it's about 67% efficacious against a variant, the efficacy is a lot more in an individual, because you have cell-mediated immune responses too. Now, how well it would protect depends on the kind of CMI responses and the kind of antibody responses that are elicited in individuals. So far, you can see that the RNA vaccines, like Moderna vaccines, like the Moderna, is a lot more efficacious in protecting against variants, as opposed to the adenovirus-based vaccines, or or maybe the heat-killed or protein-based vaccines. Is an important reason why. These RNAs make proteins within the cells, and these proteins need to be made within a cell to elicit a very good CMI or a cell-mediated immune response. Whereas if you give a protein directly into the bloodstream, it does not enter a cell. So it's coming from the outside of the cell. So that kind of a cell-mediated immune response that's elicited for a protein-based vaccine is not as broad, or I would say as it's fairly different compared to a protein which is being produced inside the cell, inherently. A DNA vaccine now, importantly, now would also produce the protein inside the cell. I mean, the DNA is made into an RNA, and then a protein is made inside the cell. So we're hoping it would be a lot more, efficacious against multiple variants. But of course, that being said, if you find variants coming in, it's very simple to make a new DNA vaccine with the variant protein. Now, in the case of SARS-CoV-2, it would be the spike protein. That's where all the variations are, which makes it infect. So it's very, very simple to synthesize a new DNA with spike protein rather than grow it up and make fresh protein. It's very, very easy to just make few nucleotide changes, get a new plasmid, and again immunize people.
Yeah, but that would mean that you will have to bring them in again and apply this pressure gun.
Right, and you don't even need to bring them in. This is the beauty of the DNA vaccine, because it's stable. People can go house to house.
You can take it to the people. Okay.
You don't need to have any sterile conditions. You don't need to have a big cube. You can potentially take this loaded, kind of an instrument, go house to house and immunize, or in schools. It's relatively exceedingly easy to do.
That is correct. How many doses would need to be given in this case? Is it the same two or?
I must say it depends what they have tested right now. I think they've tested a 3 milligram DNA dose given 3 times over a period of 28 days apart. So over a period of roughly 3 months, I think they've, or is it a 2 milligram dose, something like that. They've given a 3 dose, but now they're actually trying out a 2 dose too, where you can just go in for a double dose of 3 milligrams, I think. So depending on the amount of DNA and how it works, you can tweak it. I would think that you definitely do require booster shots, particularly because the efficacy of DNA vaccines are not as great as
because of that
viral vaccine. But you can improve upon it by increasing the amount of DNA, provided there's no toxicity. But again, given the fact that this is not painful and it's something very easy to do, booster doses should absolutely be no issues at all. You don't have to book and wait and rush at the time. I think it's easily administrable.
Okay. Is it also available? Would it be available for children as well? Is there any age bar or age limit or something?
See, that's very interesting because I don't think people have tried it in children. But what's nice is this particular vaccine that's now being brought up in India has actually been tested in a cohort which also had 1,000 so-called pediatric cases ranging between 18 and 24 years of age. So that's like the lowest age that's been tested in the USA too. And in the USA, you may or may not know, when individuals in this age range got the mRNA vaccine and even lower, some of them had real adverse effects of inflammation because of the RNA. So there was an exceedingly high level of cardiac issues that developed temporarily in kids, which is now clearly been linked to the RNA vaccine because they are strongly inflammatory. It's not, I don't think it was seriously adverse or anything like that, but you could see it because kids are prone to having a higher inflammation status compared to adults because they're exposed to a lot of antigens. There were some that did experience some level of cardiac issues, and a few were in fact hospitalized, I think, for some time, but that was the kids. They claim that the DNA vaccine doesn't do that and was found to be very safe and tolerated in individuals between 18 and 24 years of age.
So it's actually safer than the mRNA
possibly
in a way.
Safer. I would be very cautious. It's possibly safer.
Is it possible for the patients to get infected even after they have been injected with the DNA? Because there have been cases with mRNA as well.
So I would like to clarify 2 things here, which I think is important. Many people fear taking a vaccine because I heard this statement from many people that I've interacted with in India who've come and spoken to me when I've given talks, saying that I've heard people get symptoms of COVID even after the vaccine. So the thing to bear in mind is the symptoms of COVID after vaccine is not because of the vaccine. It's because it takes a period of time for the immune response to set in. So if the vaccinee comes in contact with somebody who's having infection, they will get infected until the immune responses come in. So the vaccine is essentially a very small portion of the virus. It's just one protein of the several it makes. And this protein cannot cause disease like the virus. So the symptoms you see in a patient of COVID-like symptoms are not because of the vaccine, but because they have come in contact with the actual virus from another infected individual. before they developed enough immune responses. That being said, yes, it takes time to develop immune responses. It doesn't happen overnight. And several cases, in fact, have gotten serious disease because of exposure to COVID-infected patients, SARS-CoV-2 infected patients, especially after the first dose of the vaccine, because you do need a booster dose to get, a robust immune response. So until the robust immune response sets in, which may be after the second dose in the case of viral vaccines, or it may be after the third dose in case of a DNA vaccine. Yes, I mean, if an individual or a vaccinee comes in contact with an infected individual, there are high chances that they can contract the disease. So you have to mask up and really stay safe until you know that you've taken the required dose and for the duration of time that's been that's been advocated by the manufacturer and the medical agencies.
Okay. Now, one last question here, that a person who is vaccinated and he has spent the amount of time which is guaranteed by the vaccine manufacturer, would he still be asymptomatic spreader, to say the
Absolutely, yes.
Oh, okay. So masks are here to stay.
See, what happens is all your immune responses in humans as well as in animals, non-mammalian responses, wane with time. So you know that, it doesn't mean that if you, I'm just going to take a sip of water.
Please.
So what happens is with vaccines, with any kind of immune response, the body doesn't need to maintain that immune response for the life of the individual. It's at the peak when there's a virus, and then the immune response goes down and becomes memory response. So next time when you see the virus much later, you have memory cells in your body that remember the virus and again start an immune response. It takes time to build up. So what happens is the memory response comes back soon enough. So a vaccine would probably not have symptoms of disease, but they can still get infected for a short period of time. It will be very mild symptoms. It will not go to the hospital. It will not become severe disease. So it will be like catching a cold. Like a flu shot you get every year, you will get a very mild cold or not at all, maybe if you have a very robust immune response, but you are able to fight off and it doesn't go to a serious stage. But during that phase of infection, you can potentially spread it. It won't be as bad as a person who's not vaccinated, because in a person that's not vaccinated, the viral titers go really high. Whereas in a person who has been vaccinated, titers will probably not reach that level that you see in a non-vaccinated individual. But yes, a spread to a non-vaccinated individual can happen. So people who are not vaccinated should wear masks, while people who are vaccinated can potentially, if it's safe enough, remove masks. But I would advocate wearing masks even now, given all the variants everywhere.
Correct. Because if this DNA vaccine actually takes care of the variants, it takes care of children, and it also is going to be maybe 60 or 65% efficacious, then that perhaps is the solution for a country like India, where masses need to be vaccinated very quickly. Because if we delay, that means we are allowing the disease to spread much more and more that way.
Absolutely, both from a commercial perspective, from the
operational needs,
and the mass scale that's required in a place like India. And, I think DNA vaccines is something that it should definitely be an option. At this stage where there's very little else available.
That's a fact, correct. If everybody had mRNA vaccine, then perhaps we wouldn't be talking this thing.
Absolutely.
Because there is shortage and there are more and more people need to be done quickly. I think time is the essence here.
Time is
much faster.
You can also deal with variants coming in quickly if it is something that's efficacious. It's easy enough to modify this vaccine to meet the demand of new variants.
So that seems to be the order of the day, and looks like that is the answer to a prayer of 1.34 billion people, 134 billion people here. So probably that is what we are looking at. And thank you very much for giving all this information, because I could understand all of it, almost all of it, and I am a novice in this particular subject. So I'm sure our listeners would be very happy and will be that much enlightened with this talk. Thank you very much for your time and for joining this interview, ma'am. It has
Thank
been
you
a pleasure to talk with you.
It's a pleasure to speak to you.
Yeah, thank you. And all the best. Thank you once again.
Thank you very much. Thank you.
Yeah. Okay, bye.